Hello guys, welcome to my channel. So as you know, we are discussing about the protein function, and in that we are discussing about the immune system, right? So if you saw the last video, we discussed about the introduction of this immune system, the cellular immune system and the humoral immune system. And so moving further in the topic, as we know, like each recognition protein of the immune system, either a T cell receptor or an antibody produced by a B cell, binds some particular chemical structure, distinguishing it from a virtually from all others. So humans are capable of producing more than 10 raised to 8 different antibodies with distinct binding capacities. So how much? 10 raised to 8. So humans are capable of producing 10 raised to 8 different antibodies with distinct binding specifications or specificities. So given this extraordinary diversity, any chemical structure on the surface of a virus or any invading cell will most likely be recognized and bound by one or more antibodies, correct? So antibody diversity is derived from the random reassembly of a set of immunoglobulin gene segments through the genetic recombination mechanism, right? But like a specialized lexicon is used to just describe the unique interaction between antibodies of for example or a T cell receptors and the molecules they bind. So any molecule or pathogen capable of eliciting an immune response is called antigen, right? So that elevates the immune response, so that are called antigens. All right, so I will write down the definition of this antigen. So what is it? Any molecule or pathogen, right? Or pathogen capable of eliciting the immune response is called antigen. It's called an antigen. So this is an antigen. So an antigen may be a virus, a bacterial cell wall, or an individual protein or other macromolecule, right? So antigen can be anything. So a virus, a bacterial cell wall, or a macromolecule, or just an individual protein. So a complex antigen may be bound by several different antibodies as well. If there is a complex antigen, so they might be bound by several different antibodies. So an individual antibody or the T cell receptor binds only a particular molecular structure within the antigen, and that is called an antigenic determinant or an epitope, right? So an individual right, an individual antibody or the T cell or the T cell receptors binds only to the particular molecular structure within the antigen, and what they are called? It's antigenic determinant or the epitope right? So this is what it is called. So an individual antibody or T cell receptors that just bind only a particular structure, molecular structure within the antigen, that is called an antigenic determinant or an epitope, right? So it would be unproductive for the immune system to respond to a small molecules that are common intermediates or the product of the cellular metabolism. So the molecules of molecular weight, like for example less than 5,000, are generally not antigenic. However, when the small molecules are covalently attached to the larger molecules or larger proteins in the laboratory, right? And remember, in the laboratory, they can be used to elicit an immune response. So these small molecules are called haptins. They are called So this is the term. Here's Haptins, right? So what? So there are just a small molecules that are covalently attached to the large proteins in the laboratory. They can used to be elicit an immune response or elevate an immune response, and these molecules are called haptins, right? So the antibodies produced in response to the protein linked haptins will then bind to the small molecules in their free form. So you got it. So like first how the antigen is being produced. So the small molecule is linked first to the larger protein. So when it is linked to the larger protein, this will elicit an immune response. So that will produce an antibody, right? So now the antibodies is produced in response to the protein that is linked to the hapten, that is linked to this small molecules. So will then they will bind to these same small molecules in their free form as well, as they were attached to this large molecule, and because of that the antibodies is produced. So even when the small molecules come without the larger protein, the antibodies can still bind to the same small molecules in the free form. So such antibodies are sometimes used in the development of analytical test, right, or as a specific ligand in the affinity chromatography. So now we'll see, what we'll learn is antibodies have two identical binding sites, two identical antigen binding site, we'll say that. Right, antibodies have two identical antigen binding sites. So immunoglobulin G, or it's called IgG as well. So write it down. Immunoglobulin G or IgG. So this is the major class of antibody molecules and one of the most abundant proteins in the blood serum. So this is the IgG is the major class of the antibody and one of the most abundant protein in the blood serum. So this one of the most abundant protein in the blood serum. Blood serum. Right, so the IgG has a 4 polypeptide chains, right? So IgG has 4 polypeptide chain. So two the large ones that are called two heavy chains, right? So write that down. Large one that are called heavy chain, V is 2, and there is 2 light chain as well, right? Okay, so this 2 heavy chain and this 2 light chain are linked by non-covalent and disulfide bond into a complex of molecule that is the molecular weight is greater than or equal to 15,000, right? So this both are linked by non-covalent and disulfide bond. Non-covalent disulfide bonds. Right, so the heavy chain of this IgG molecule, what it does, it interact at one end, then branch to interact separately with the light chain, forming a Y-shaped molecule. So I will show that. Right, so how it does. So make it here, I think you will see that. All right, so for example, so like so just need, so for example, so H is represented by the heavy chain, right? So it will, so this heavy chain will branched here, right? And down here, and this will interact with the light chain. This will Right, so something like this it makes. So the heavy chain of this IgG, so this H is represent the heavy chain that will interact at one end. So they both will interact at one end, right? And then they will branch it to interact with the separate light chain. So they make like this, right? They both separate, and then they break it and separate with the interact with the light chain, forming a Y-shaped molecule. So this is just a rough image of the IgG molecule. All right. So at the hinges, so why we draw this picture? So for example, this is the Y shape, and here that's the hinges of this, right? So like it can turn like this or whichever. So like for the door has a hinges, so like this Y shape has this hinges here. You can see this part of the center here. So at the hinges separating the base, so this is the base of an IgG molecule, from its branches, this immunoglobulin can be splitted with the proteases, right? So splitting with the proteases, that is pepin. So that's an enzyme. Right, so what happens? So when this is splitted with the protease pepin, what it will liberate a basal segment. So this is the basal segment, this is the lower portion. So that is called FC, right? Like this written as, because why? Because it usually crystallizes readily. So this basal segment crystallizes readily, that's why it is splitted first. And then the two branches, so these two branches here and here, so that is called FAB. So that's why the two branches, that is called the antigen binding fragment, FAB, right? And each branch has a single antigen. This both has a single antigen binding site. So when it is treated with this protease enzyme, so this will get splitted, the basal segment will get separated, and the two branches, that is called antigen binding fragments, also get separated. And this both has the single antigen binding site, right? So this was the IgG. So the fundamental structure of this immunoglobulin was first actually established by Gerald Edelman and Rodney Porter, and each chain is made up of an identifiable domains. So if you remember the definition of domain, so domain is like when there is like large proteins like this, so each chain is identified as a domain or can be as a separate entity, right, other than the protein. So each side of this, so that's the domain. If you don't remember, you can see the previous video, we discussed about domain and motifs. So each chain is made up of this, each chain, the two heavy chains and the two light chains, they are all separate entity, they can act as a separate entity, so they are identifiable domain. So some domains are constant in sequence, right, all four of them, and structure from one IgG, so they are constant in sequence and the structure from one IgG to the others to the next one, and some are variable as well. So the constant domains have a characteristic structure that is known as immunoglobulin fold, right? So there are like three of these constant domains in each heavy chain and one in each light chain. So the heavy and the light chain also have one variable domain on each, in which most of the variability in the amino acid sequence is found. So the variable domain, why it's variable? The variable domain associated to create the antigen binding site, allowing formation of an antigen antibody complex. So for example, so this is our IgG. So when antigen comes here, for example, So what will happen? So just a rough diagram. Something like this. So, alright, so the antigen when it comes, so it will, so this is the 2 antigen binding site. So that's AG, I will just write AG for the antigen. Right, so this is where the antigen will bind. So these are the 2 antigen binding sites. Right, so in many vertebrate, IgG is one of the 5 classes of immunoglobulins. So each class has a characteristic types of a heavy chain. So like it's denoted in a Greek symbols like alpha, theta, gamma, lambda, mu. But the different types of 5 classes of immunoglobulins are, so the one that we studied, IgG, right? Then IgD, then IgE, right? And TA and IgM. So these are the 5 classes of immunoglobulins, right? So like this all have a heavy chain. So the heavy chains are represented, as I mentioned, in the Greek symbols like alpha, theta, gamma, mu. But the light chains are just as denoted as a kappa and lambda that occurs in all of the classes of immunoglobulin. So the overall structure of the IgD, this is IgD. So IgD and the IgE are similar to the IgG. So this 3 has overall structure the same as the IgG. Right, so IgM can occur as a monomeric membrane bound form, or also can be in the secreted form, that is cross-linked pentamer of their basic structure. So like, so on your screen you can see the picture of this pentamer IgM. IgA found principally in the secretions such as saliva, tears, milk, and can be monomer, dimer, or trimer as well. So where the IgA is found? In secretions such as saliva, tears, and milk. Right, so IgA is found in secretions such as saliva, tears, and milk, and their structure can be monomer, dimer, or trimer. IgM is the first antibody to be made by B lymphocyte and the major antibody in the early stage of primary immune response. Right, so that's important. So IgM, what is the IgM? Is the first antibody to be made by B lymphocytes, and also the major antibody in the early stage of primary immune response. All right. So IgA found in secretions, tears, saliva, milk. IgM, that's the first antibody made by the B lymphocytes and the major antibody in the early stages of primary immune response. So some B cells soon begin to produce IgD with the same antigen binding site as the IgM produced by the same cell, but particular function of IgD is less clear. The IgG that we studied before, this one IgG, that's a major antibody in the secondary immune response, which are initiated by the class of B cells that are called memory B cells. So the IgG comes in the IgG is the second stage of immune response, right? Immune response, and these are initiated by the class of B cells that are called memory B cells, right? So as a part of organism, ongoing immunity to antigens already encountered and dealt with, IgG is the most abundant immunoglobulin in the blood. So when IgG binds to an invading bacterium or virus, what it does, it activates certain leukocytes such as macrophages to engulf and destroy the invaders, and also activate some of other parts of the immune responses. So receptors on the macrophage surfaces recognize and bind the FC region of IgG, and when these FC receptors bind an antibody pathogen complex, the macrophage engulfs the complex by phagocytosis. Right, so on your screen you can see the picture that we discussed how macrophage engulf the FC region of IgG that is attached to the virus, right? So this is how phagocytosis is done. Right, and the antibodies bind tightly and specifically to antigen. So just to know, the binding specificity of an antibody is determined by the amino acid residues in the variable domains of its heavy and light chain. So these are the different kinds of chain, and each chain represent one domain. So many residues in these domains are variable, not equally so. So some, particularly those lining the antigen binding site like this, are hypervariable, especially likely to differ. So the specificity is considered by chemical complementary between the antigen and the antibody, right, and its specific binding site in terms of like shape and the location of charge, that is non-polar or polar and hydrogen bonding groups. So like for example, a binding site with a negatively charged group may bind an antigen with a positive charge in the complementary position. So in many instances, complementary is achieved interactively as the structure of antigen and the binding site influences each other as they clump close together. So the conformational changes in the antibody and or antigen then allow the complementary groups to interact fully, and this is an example of induced fit. So that's the complex of peptide derived from the HIV model and a PAB molecule that we see here illustrate some of these properties, right? And the antigen and the antibody interaction is actually the basis for a variety of important analytical procedures. So the extraordinary, actually the binding affinity and the specificity of antibodies make them valuable analytical reagent. Two types of antibody preparation are in use, that is polyclonal and monoclonal antibodies. Right. So first, polyclonal antibodies. Right. What is so polyclonal antibodies are those produced by many different B lymphocytes responding to one antigen, such as protein injected into an animal cell or into an animal. So that cells in the population of B lymphocyte produces antibody that binds specific different epitopes that we see here, different epitopes within the antigen. So thus the polyclonal preparation contains a mixture of antibodies that recognize different parts of the protein. So vice versa, monoclonal antibodies. So that means it binds to just one side. In contrast, are the synthesized by a population of identical B cell grown in the cell culture. These antibodies are homogeneous or recognizing the same epitope. Right, and this technique are for producing monoclonal antibodies are developed by George and Caesar Milstein. Right, and there are like exploited analytical techniques such as ELISA and immunoblotting. Right, so ELISA is, these are the two different techniques that are used. So this is enzyme linked, the full form is enzyme linked immunosorbent, right, ELISA, and the another one is just immunoblotting or also known as Western blot. So these are the two techniques, that is the exquisite binding specificity of immunoglobulin for the analytical techniques, right? Alright guys, so that was the immunoglobulin topic. Right, so we'll see you in the next video. We'll still continuing in the protein function as we are going through biochemistry. So we are discussing more about the molecular level rather than the functional level, but we will just mention the functions are just a simple thing, right? So that's why we are just seeing the molecular level as well. All right, thank you guys for watching, and don't forget to subscribe my channel. Thank you. We'll see you.